This lecture covers microbial genetics of gene expression. Obviously, microbes have a very rich array of gene expression mechanisms that can occur at the level of transcription or translation. So I'll try to get into a little bit of both, with the emphasis being on transcriptional gene regulation. One of the primary components of, or mechanisms rather, of gene expression at the transcription level are two-component regulatory systems. And they're predicated on a phosphorylase system. And so these are systems that are used to turn genes and entire operons on or off in response to environmental conditions. So not surprising, microbes have to really have to modify gene expression rapidly to adjust for rapidly changing environments. So, if you're a host-associated microbe and you suddenly are sneezed or coughed onto a hand or on a lab bench or whatever, suddenly the environment has changed significantly, and obviously the microbe has to then make adjustments very quickly in order to survive that catastrophic change. And one of the ways they do it is through these two-component regulatory systems. So the prototypical two-component system is outlined here, and this is a mechanism that was worked out in E. coli, and it's the regulation of porin proteins using a two-component system. And so the main porin proteins in E. coli are OMP-C, which is a smaller pore porin that the microbe would would definitely favor in the presence of a more concentrated environment. And as a result of being smaller pored, it allows fewer of these ions to come into the cell. Obviously, if you're allowing too many ions in, you can create this imbalance across the membrane. And if it's too concentrated here, water will flow in and the cell will lyse. The other porin in this particular model is OMP-F, which is a porin that's favored in dilute environments. It's a larger pore porin, so it can allow more things to come in. So imagine in this hypothetical situation here that we're in a high osmolarity environment, and therefore we would want to favor OMP-F expression over OMP-C. And so the proportion of OMP-F you want to be higher than OMP-C. Now, in this particular case, as the high osmolarity environment seeps into the periplasm here, that affects the periplasmic side of the first component of the two-component system, called MZ for envelope Z, generically referred to as the sensor kinase in this particular model. And so in this particular case, the cytoplasmic end of the sensor kinase is phosphorylated. So ATP gives up a phosphate to this crucial histidine residue here in the cytoplasmic end of MZ. The phosphate is then transferred to this aspartate residue in the second component of the system, OMP-R, the so-called response regulator. So OMP-R phosphate can then act as a repressor, can basically sit on the operator within the promoter of OMP-F and turn it off. Remember that this is a high osmolarity environment scenario, and so we want to favor the expression of OMP-C and cut down on the amount of OMP-F that is produced in the progeny cells. OMP-R phosphate will interact with the promoter for OMP-C and actually increase expression of OMP-C. And so again, simultaneously repressing OMP-F and turning on OMP-C. So it has a dual function as both a repressor and an activator. The other thing that OMP-R phosphate does is it activates the expression of a small RNA called MIC-F. And MIC-F is, again, as I said, is an antisense RNA. And so here's our antisense RNA from the MIC-F gene, and here's our OMP-F messenger RNA. And so there's a region here of homology near the 5' end of OMP-F. And as a result, any bits of OMP-F that were expressed before OMP-R phosphate turned it off, so any bits of transcript, transcript that were produced here would be turned off. So that's a fine-tuning mechanism as well, just so that, again, so that the cell, your progeny cells, don't have as much en peth. So again, it's a double layer regulatory mechanism. Another cool example of a two-component system is Enterococcus, the so-called VRE, vancomycin-resistant Enterococcus species. And these guys have glycopeptide resistance specifically to antibiotics like vancomycin, which is a cell wall synthesis inhibitor. And so vanco present in the environment will activate this response, this sensor kinase, VAN-S. And so by activate, I mean it causes VAN-S to autophosphorylate, just like you saw in the OMP model. And so just like you saw with MZ. So VAN-S autophosphorylates, and then the phosphate group is transferred to the response regulator VAN-R. And then VAN-R phosphate can activate the VAN operon that you see here, H, A, and X. And these proteins, the proteins that are encoded by this DNA, have the function of blocking the ability of vancomycin to bind to peptidoglycan. So again, just to remind you, imagine this is a growing peptidoglycan chain, and here are the cross links. So here's the uprights, sort of the ladder, alternating NAM and NAG residues linked by a glycosidic bond, and then the pentapeptide side chains that come off of the NAM residues. And obviously the microbe, in order to polymerize this and strengthen it, the microbe has to kick off this alanine, kick off one of these terminal alanines here, and then form a linkage between the adjacent base, between the adjacent amino acids. So you see there's and then kicks off that alanine. And so what and obviously the job of vancomycin, for example, the antibiotic vancomycin, is to sit right here on these terminal alanines to prevent that cross-linking. And so by preventing that cross-linking, you have a weak cell wall that the bacteria will then be unable to strengthen, and the cells will fall apart. And that's basically the mechanism of action of vancomycin. It's very similar to the mechanism of action of penicillin as well. But the point is, by the VAN operon that I showed you modifies this terminal alanine so that it, as I show you here, becomes a lactate. So VAN-H converts the pyruvate to lactate, and then VAN-A, the protein VAN-A, will bind one of the second to last alanine to the lactate that's made here. And so in that process, VAN-X also cleaves, cleaves that terminal alanine off, and then the lactate is applied to that terminal alanine by the VAN-A protein. Anyway, the point is it modifies the target that vancomycin would be looking for, and because the target's no longer available, vancomycin doesn't work, and so you get these microbes like VRE, the vancomycin resistant enterococci. This is I'll look at the Toxar regulon, and so I wanted to zoom out a little bit. You already understand, hopefully, the concept of an operon, in which you have here's a mini operon right here, in which you have a promoter that regulates expression of, a single promoter that regulates expression of 2 or more genes. So this little mini operon here in Vibrio cholerae is essential for creating a membrane responsive protein system for allowing cholera to respond to the environment. And so again, an operon, a single promoter that regulates expression of 2 or more contiguous genes. But a regulon is a bunch of genes and operons that are all keyed into the same circuit and co-regulated, and that's what's pictured here. So obviously the regulon and operon mechanisms are really important for microbes to be able to rapidly respond to changing environment, rather than trying to flip a bunch of different switches and that are not close to each other. Imagine yourself standing at a big switchboard and you've got switches all over the place, and if something changes, it's not very efficient to have to run all over the place and try to flip a bunch of switches. It'd be nice to have one master switch that is keyed into a bunch of different a bunch of related functions. And so in this example, the master switch is TOXR and TOXS from the TOXRS mini operon here. And so these encode proteins, TOXR, which exists as a dimer in the inner membrane, the cytoplasmic membrane of Vibrio cholerae, and then TOXS, which kind of straddles these proteins and sticks into the periplasm of this gram-negative bacterium. So here's our outer membrane, and then the cell wall would be right in here. And here's the periplasm, obviously. And so some environmental signal, temperature, osmolarity, the composition of the medium, for example, is there bile present? These are all telltale indicators that the bacterium has gone from swimming around in a mud puddle or an estuary to suddenly finding itself in your duodenum. And so these environmental signals that say, okay, I'm now in a human body, will activate these TOXR and S molecules, these proteins, in order to activate a series of gene expression that would trigger the microbe to adapt to life inside the host. And so one of the first things that happens is these TOXRs monomers dimerize. So imagine these just kind of crossing over a little bit. So let me back up. TOXS perceives one of these types of signals, or a combination of these signals, and that causes TOXR to dimerize. In its dimerized state, it can actually physically interact with the promoter, for example, of the cholera toxin operon. So it can activate, that's what these little pluses are, can activate the cholera toxin operon. There's no reason for Vibrio cholerae cells to be pumping out cholera toxin while they're swimming around in an estuary, for example. So it's only when the cells are inside the duodenum do they really need to pump out cholera toxin. The other thing that this TOXR-TOXS interaction does is it activates expression of TOX-T over here. TOX-T is itself a transcriptional activator, which can be synthesized and activates the expression of this toxin co-regulated pilus. So the toxin co-regulated pilus does a bunch of things, like, for example, helps cholera cells stick together and clump up, which is an adaptation for survival in the host. And so again, and then TOX-T also activates expression of more cholera toxin. And there's other inputs as well, like TCPPH, which is a transmembrane protein, and these transcription factors, APHA and APHP, all these kind of feed in. But I want you to look specifically at TOXR, TOXS, activating CTXA and B, and activating the promoter for TOX-T, which in turn activates the toxin co-regulated pilus, and in turn activates even more cholera toxin production. So cholera also has a quorum sensing cascade that's very similar to one that I'll show you in Vibrio fischeri that we've worked with a little bit. Now there's some differences obviously, but many homologs. The parallel systems that you see here are the CQS system, cholera quorum sensing system, and then the autoinducer 2 system. A lot of microbes have this one. This is more of like a generic system that allows microbes to kind of communicate with each other across species. And this one is more species specific. And so in this particular case, you have it's basically a multiple component relay system. It's not a 2-component system because there's an additional component, but so we can maybe call it a 3-component system. In this particular case, the CQSA is an autoinducer synthase that makes these quorum sensing molecules, these little diamonds here. And then LuxS is an autoinducer synthase that makes these triangles. But imagine in the absence of these diamonds or these triangles, the sensor kinase that you see here, CQSS, will autophosphorylate, then transfer the phosphate group to this response regulator called